Hello students, how are you? I hope you all are fine. Today in this part we are going to discuss about the microbiology. See, it is a very well known branch under the applied biology. Okay, so today we are going to discuss some important points under the microbiology and what we are going to study in this microbiology that we'll see here. So, we'll give the title, that is microbiology. Okay, so before understanding various concepts, see what concept that we are going to discuss in this chapter. I'll highlight these concepts. Okay, but before that we should understand what is meant by microbiology. So microbiology is nothing but it is the applied branch of biology, or it is the branch of biology that deals with the study of microorganisms. Okay, so when we talk of the microorganisms, these microorganisms are the living organisms which are very minute and which cannot be seen by our naked eye. Okay, so studying such living organisms, okay, is through the microbiology, or it is called as the microbiology. Okay, because their size is very minute, they are studied, their structure, okay, they range under the micron, so we call them a microbes, okay, or the microorganisms, and their study is called microbiology. So you can write the study of microbes, or the study of minute living organisms, study of minute living organisms, okay, study of minute living organism which are, or which cannot be seen by our naked eye. Okay, so see, the study of minute living organisms, and these minute living organisms can be referred as the microbes, okay, which cannot be seen by the naked eye, is known as microbiology. Okay, so in the field of microbiology, various different scientists has given their contribution. Okay, and microbiology is not about the single organism or single living organism, it contributes different types of microbes. Now what is meant by microbe? Microbe, or it can also be called as the microorganisms. Sorry, microorganisms. Okay, so microbe or microorganisms generally, okay, it is referring to those organisms which are unable to see by the naked eye, right, or their size is very low. Okay, so that we have to observe this living organism with the help of particular devices such as the microscope. Okay, through which, or what is microscope basically? It is the uh, it is a device consisting of series of lenses, okay, series of lenses which help to magnify the minute images. Okay, so whatever images we are we are getting onto the slide, that will be magnified, and those magnified images may be of the minute organisms, and these are the microorganisms. Now microorganisms are of different categories. If we talk of the different microorganisms, we have also discussed during the kingdom Monera, right? So here, like according to Whittaker's 5 kingdom classification, okay, or the classification system includes different category of the microorganisms. So let's write down the categories of the microorganisms, like bacteria, then viruses, then protozoans, then fungi, etc. So these are the major categories of the microorganisms. Okay, so here you can see the microorganisms such as bacteria. There are different types of bacteria are seen in the globe. Okay, as well as viruses, protozoans, and fungi. So when we talk of the bacteria, we have a specific cell structure. They are generally referred as the prokaryotes. Okay, in case of viruses, they are referred as acellular because they are considered as obligate parasite. So that means they require host for their multiplication. Then next, protozoans. If you talk of the protozoans, okay, some of the protozoans or they are included under the protist. Okay, kingdom protista. So here protozoans, they may be unicellular, okay, or acellular again. So depending upon their properties, we can differentiate them. Then fungi, this can be the unicellular, multicellular, or filamentous type of living organism which may reproduce by sexual or asexual means. So here all these sexual and asexual means of characters can be seen in all the different types of microorganisms. Now here in this chapter we are going to discuss few important points that we'll note down right now. Okay, so in microbiology we will be talking about the disasters. Okay, what are the disasters, different types of disasters. Okay, and then we'll discuss about the we'll note down the points. Okay, we'll highlight those points and then we'll start with step by step. So this will be the definition. Okay, and there is a huge scientific contribution in the field of microbiology again. So in this chapter, the points to study are points to study. Okay, now what points we are going to study in this chapter is number 1, disaster management. Okay, so this disaster management will discuss in the end of the chapter. Okay, so we will start basically with the basic concepts of microbiology. Okay, and then we will discuss about the different microorganisms, their activities. Okay, where they are involved, what are the different functions of this microorganism. Okay, where they are used basically. Okay, so that we will discuss here. Then next, another point, that is cell structure. Okay, then already we have discussed about cell structure also in previous classes, we have discussed about the prokaryotic cell structure, we have discussed about the fungal cell structure. Okay, we have also discussed about the protozoans, what type of cell structure they have and what type of mode of nutrition, mode of reproduction they show. Okay, so just we have to revise all these points over here. Okay, then another point that we are going to discuss is the nature of microbes. Nature of microbes. Okay, so what kind of nature they have? See, in case of bacteria, we can differentiate them into gram positive and gram negative bacteria. Okay, apart from that, there are different types of differential staining is included under this bacteria. So what we can differentiate the bacteria like acid fast bacteria are there. Okay, so apart from that, in case of fungi also, in case of protozoans, okay, we can differentiate them into different categories. The next, harmful and useful bacteria. Harmful and useful bacteria, or sorry, microorganisms. Microorganisms. So in this part we will talk about the harmful effect as well as the useful effects of the microorganism, how they are beneficial for humankind, okay, or for human welfare, that we will discuss here. Then next, we will discuss about pathogens. Okay, then we will discuss about the environmental management. Okay, then we'll discuss about the industrial microbiology. Then we'll discuss about the dairy industry. Okay, dairy microbiology. Then we'll discuss about another point, that is probiotics. Okay, probiotics and uses of probiotics. Then next, microbes or microbial enzymes. Next, microbial enzymes. Then we'll talk of the biofuel, and pollution control. Okay, biofuel and pollution control. Biofuel and pollution control. Then we'll discuss about the clean technology. Then we'll discuss about the microbes and farming. So students, these are the sub points that we are going to discuss in this chapter, that is about the disaster management. Then we'll discuss about the cell structure. Okay, in cell structure we'll discuss about the what type of cell structure they have, whether they are flagellated, they are non-flagellated. Okay, what type of cellular composition they have, cell wall composition. Okay, that already we have discussed in the cell chapter, but still for an overview we will discuss here. The next, nature of the microorganism, what type of nature they have. Okay, means in what type of climatic conditions they are been grown and what type of nature they exhibit. Then whether they are harmful or useful microorganisms. Then pathogen. What is pathogen? That is the disease causing organisms or the disease causing microorganism is referred as the pathogen. So here we will discuss about the pathogens. Then we will move towards the environmental management. Environmental management is somehow associated with the disaster management. So we will combinedly discuss about the disaster and the environmental management. The next, industrial microbiology. In industrial microbiology, we will talk about the different industrial products. Okay, there are different industries such as textile industry is there, pharmaceutical industry is there, food industries are there. Okay, detergent industries are there, like beverages industries are there. So different, even though organic acid production industries are there, all these industries, they are using the microorganisms for better yield of the product. Okay, that how they are yielding this, that we will discuss during this. Then dairy microbiology. Dairy microbiology is that it is dealing with the processing of the dairy products. You know, in shops also or in the ice cream parlors, we get different variety of ice creams, right? As well as different dairy products are available, like yogurt is there, okay, Shrikhand is there. Then we have what we say, ice creams are there, right? All these products, how they are manufactured? They are manufactured with the help of certain bacteria, and these bacteria, they are already present in the milk, right? So what we are doing, we are culturing this bacteria in larger amount so that they will ferment the milk and they will produce the useful product like curd, and from curd we are producing the butter as well as the buttermilk is there. Then we are producing the ghee. Okay, so many different useful products are produced with the help of microorganisms. Then we will discuss about the probiotics. Probiotics are the recent type of recent or newly formed antibiotics, you can say, or these are the supplements which induces the immunity. Okay, and these are also produced with the help of microorganisms. Then microbial enzymes. Now see, in different industries, like in the dairy industry or the other industries are there, in which different types of products are produced. Okay, like if we talk about the detergent industry, different types of detergent we have: Ariel, Tide, Surf Excel. Okay, all these detergents, they consist of different types of enzymes. Okay, so you might be seen in the advertisements also, when there is a stain on to the cloth. Okay, at that time these detergents or the industries, they show that this microparticles, they will enter within the cloth, they will remove the stains or the lipid structures. Okay, oils basically. So this, how these oils are released or been removed with the help of enzymes called lipases. Okay, so such enzymes are been produced by the microorganisms, and these enzymes are added into these detergents. Then proteases, different types of enzymes are there: lipases, proteases, nucleases. Okay, so different types of microbial enzymes can be used. Then biofuel, how biofuel is been produced with the help of microorganisms, and how it is been also implemented in the pollution control. If we talk of the pollution, different types of pollutions are there: air pollution, water pollution, okay, soil pollution is there. So all these pollution, they can be controlled with the help of the microorganisms. Okay, so microorganisms play a great role in what we say, protecting our environment also. Okay, then clean technology, again it is associated with the pollution control. Okay, then microbes and farming. So yes, as most of the countries, they have a base, that is agriculture, and agriculture, it is also associated, it is directly associated with the microorganisms. See, different microorganisms in the agriculture field, they are responsible for uh, providing different types of functions. Okay, if we talk about the growth of the plant, see, if it is concerned of the growth of the plant, what is required for growth of the plant? We require a nutrient that is called nitrogen, and how this nitrogen is been made available to the plants with the help of nitrogen fixing bacteria. Okay, because nitrogen have different forms, like ammonia is there, sometimes it is in the form of urea. Okay, but that urea or ammonia, or sorry, urea or other like uric acid will be there. So all these other components of the, or the other forms of the nitrogen, are not directly available to the plants. For making this available to the plant, there are certain bacteria or microorganisms are there, commonly called as nitrogen fixers. Okay, so what they do, they fix the atmospheric nitrogen into the plants, so that plants, they show the growth. And as we know, nitrogen is the chief metabolic component in most of the cellular organelles. If we talk of the chloroplast again, okay, so chloroplast consists of chlorophyll pigments, and chlorophyll pigment consists of nitrogen at the centermost part, or the centermost what we say, molecule into that structure of the chlorophyll molecule. Okay, so here it help in the photosynthesis also. Okay, so here there are different role, again for production of the nucleotide, there is a presence of nitrogen. Okay, so nitrogen is very important. Apart from nitrogen, we have different minerals also, which are very important. Then these are the different points that we are going to discuss here. Now here we will start with the cell structure and nature of microbes. Okay, then we will discuss about the harmful and useful microorganisms, and then we will move further with the different points. Okay, I hope you are getting this. Now let's move further. Okay, we will start with the cell structure. Okay, this point, that is the disaster management and the environmental management, will be discussed later. Okay, as these are connected to each other, we will discuss about it later. So let's start with the basic concepts in the microbiology. Okay, so okay students, so now we will move with the another concept, that is the, or the main concept in the microbiology is with the cell structure. Okay, see, basically when we talk of the cell structure, different types of structures can be found in these living organisms. Okay, some may be oval, some may be rectangular, some may be rod shaped, some may be square, or any kind of cell structure can be seen in the microorganisms, as they are adapted to survive under extreme conditions, okay, as well as whatever favorable condition they have. So that's how we can differentiate these microorganisms, okay, and their cellular structures. Now if we talk of the bacteria, okay, bacteria, they are generally referred as the prokaryotes, and we know prokaryotes means the organisms which are having very primitive type of structure, okay. So pro means here the primitive, okay. So the organism which are having primitive type of structure are referred as the prokaryotes, and it includes the bacteria, okay, and the study of bacteria is referred as the bacteriology. Study of bacteria is referred as the bacteriology, okay. Then some bacteria, they can be harmful, as we have discussed. They can be harmful, they can be useful, okay. So they have the primitive structure. Now they can be flagellated or non-flagellated. Then they can be, okay, so flagellated or non-flagellated. They consist of the cilia on their surface. Then they consist of the pili, okay. Pili may be present or absent, okay. Then they have the cell wall, which is composed of the peptidoglycan, okay. So we'll write here cell wall. They have cell wall made up of peptidoglycan, okay. So they have cell wall made up of peptidoglycan, which is nothing but a composed of N-acetyl glucosamine, okay. So here it is a type of conjugated protein, you can say conjugated or carbohydrate type, like peptide as well as glycan means what we can say, like proteins and sugar, they are combined, okay. So they form a complex network into the cell wall of the bacteria. Apart from that, they have the cell membrane, which is composed of lipid bilayer, okay. So we can write here lipid bilayer or lipid bilayer, cell membrane. See, we are writing just basic information, okay. Already we have discussed about bacteria in detail in the kingdom Monera. You can refer to those points over there, okay. But here just we are highlighting these points. So what they consist of, the what the bacteria consist of basically, okay. So they consist of lipid bilayer. Now as we know, they have the primitive structure. The cell consists of cytoplasm, but the cytoplasm don't have all the cellular organelles, right. So the nucleus, it is not well developed, or it is not been surrounded by the nuclear membrane, okay. So it can be referred as this naked DNA into this bacteria, okay. Or instead of that, we can also refer as the simple type of genetic material it is found in the bacteria. Apart from this, in bacteria we have an extra chromosomal set of DNA, which is referred as the plasmid, okay, which is called as the plasmid. So that is the extra chromosomal set of DNA, which is again adding a special feature in the bacteria, as they get a resistance, okay. It is responsible for providing the resistance, as on the plasmid there is the presence of antibiotic resistance genes, like ampicillin resistance gene is there, tetracycline resistance gene is there. So in this way they get the resistance. Now let's talk of the nature of the bacteria. Now depending upon the stain they take, okay, because when we observe the bacteria, we have to stain with the particular dyes. Now for staining of the bacteria, different types of staining methods are used, okay. Differential staining, in differential staining we stain the bacteria depending upon the cell wall composition. Now in general there is a staining technique called gram staining. In gram staining we discuss about the different nature of the bacteria, whether they are having gram positive nature, whether they are having gram negative nature. Okay, so gram positive nature means there will be higher level of the peptidoglycan, and gram negative means lower level of the peptidoglycan will be there in the cell wall. So in this way we can differentiate the bacteria. Okay, now when we go towards the harmful or useful, see we are going to discuss about all the microorganisms, but we are going with the first, that is bacteria. Okay, over here. So when we talk of the harmful as well as useful effects of the bacteria, now bacteria can be harmful, yes, as many of the bacteria they are disease causing, right? So they are pathogens. What they do, see, most of the bacteria they generally spoil the food material, right? So they are involved in the spoilage of the food material, which may release certain toxic substances, and when these toxic substances they are entered within the gut of our body, okay, at that time they are responsible for causing different types of alternating reactions within the body. So these alternating reactions may cause the disease or the disorder. Some bacteria can directly involved in the disease formation, like Vibrio cholera. These are the disease causing microorganisms. Okay, means they cause the cholera. Okay, then some bacteria they are involved in the causing certain infection in the tooth. Okay, so many bacteria are there. Then some bacteria can also cause the infection, okay, to the various different parts of the body. Okay, wherever we get the infection or wherever we get the wound, there is a possibility of getting the infection by the bacteria also. So bacteria in many way they harm our body. Most probably enterococcus type of bacteria, or bacteria which are found in the body of the intestine or the intestine, they can cause the diarrhea. The bacteria like Salmonella typhi, they causes the typhoid. Okay, then many are there. Mycobacterium leprae causes the leprosy. Mycobacterium tuberculosis causes the tuberculosis. Okay, so different bacteria are there. These bacteria they have the harmful effects. Okay, apart from that we have the bacteria which are having the useful effect. Okay, so what are these bacteria which are having the useful effect? See, bacteria is also used in various industries. Let's take an example, like in dairy industry, they are used for conversion of the milk into the various milk products. Like there will be production of the ice creams, there will be production of the what we say, yes, yogurt, cheese. Then there will be production of the Shrikhand, okay, peda. All these means different types of products are produced with the help of milk, and this milk or this conversion is been associated with the bacteria, and generally the bacteria which are involved are the lab, that is lactic acid bacteria. Okay, and this lactic acid bacteria, what they do, they coagulate the milk protein, that is casein, which is present in the milk. This protein will be coagulated, and by which they will produce the useful products. Okay, so this is how the bacteria are used in the dairy industry. Apart from this, the bacteria can be used in the detergent industry. Okay, if we talk of the detergents, different detergents are there, Ariel, Tide, okay, Rin, Supreme, like that. So all these detergents, what they consist of? They consist of the enzymes. Okay, आप लोगों ने सुना होगा कि जैसे स्टेन्स आ गए और वो स्टेन्स को रिमूव कैसे करेंगे, तो स्टेन्स को रिमूव करने के लिए क्या होता है, जैसे ऑयल है, ऑयल का स्टेन होगा, जब कोई सब्जी गिर जाती है तो उसमें क्या होता है, ऑयल होता है, और इस ऑयल को रिमूव करने का काम कौन करेगा, उसमें होता है क्या, लिपिड्स होते हैं, तो लिपिड को रिमूव करने के लिए लाइपेजेस, ओके, जैसे हम लिख सकते हैं यहाँ पे एग्जांपल्स, ओके, so we'll write here. So lipases, okay, lipases किस लिए यूज होते हैं, for breakdown of the lipids. Okay, so for breakdown of the lipids, the lipases enzymes are used, and these lipases they are added into the detergents. Apart from this, we have the proteases. Okay, so proteases they are also involved in the detergent industry for removing the stains. Okay, so different enzymes are there. Apart from this, the enzymes they are also used. Okay, they are also used in the food processing industries. Okay, in food processing industries in the sense what? See, when we talk of the fruit juices, अभी मार्केट में हम लोगों को अलग अलग टाइप के फ्रूट जूसेस मिल जाएंगे, करेक्ट? ट्रॉपिकाना इज देयर, रियल इज देयर, ऑल दिस फ्रूट जूसेस दे हैव अ क्लियर टेक्सचर. ऐसा नहीं है कि कोई फ्रूट उसके अंदर एडेड है, जैसे कहीं बीज आ गया, किसी को टैनी आ गया, ऐसे नहीं. दीज आर प्योरली क्लैरिफाइड. एंड हाउ दीज आर क्लैरिफाइड? दीज आर क्लैरिफाइड विद द हेल्प ऑफ such enzymes like proteases, okay, or the pectinases. Okay, generally in case of plant fruit, okay, pectinases. Now here what I mean to say is all these enzymes, whatever enzymes are used in the dairy industry, in the what we say, in the detergent industry, in the food industry, all these enzymes are manufactured with the help of bacteria. Okay, so different bacteria are there which are cultured under laboratory, that is called in vitro culture, and the product which are been formed by this bacteria are purified and stored in large amount. Okay, so the process called fermentation, with the help of fermentation technology we can produce the useful product by the bacteria. Okay, now here different bacteria can produce the proteases again. Okay, so in this way we can see the useful effect of the bacteria. Apart from this, bacteria can also be used in the agriculture field. Okay, for production of the biocontrol agent. Like what is biocontrol agent? See, we know that the crops, okay, in the agriculture field, they get infection through the various types of insects as well as pests, right? So to remove this insect and pest, what we do? We use the biocontrol agent, that is in the form of biofertilizers or the biopesticides. And the biofertilizers and biopesticides, they are also been produced by the different types of bacteria. Not only bacteria, some fungi are also involved, some protozoans are also involved. Okay, so different category of the living organisms are, or the microorganisms are involved in the production of the useful. Okay, now one of the emerging branch with associated with the field of microbiology or biochemistry or other fields, that is called biotechnology. Okay, it is associated with the modification in the genetic makeup of the bacteria, so that we can get a proper yield or a we get a benefits from this products. Okay, so basically what we are doing, we are fulfilling the human welfare. Okay, or we are fulfilling the requirements of the human welfare. Okay, so these are the different industrial processes in which microorganisms they are processed, and by the processing of microorganism we get the desirable product. Now if we talk of the organic acids, okay, other use can be said as the production of organic acids. Now what is meant by organic acids? See, organic acids are those acids which are produced by the living organisms, correct? So here it may include the citric acid, it may include the acetic acid. Okay, so citric acid is there, acetic acid is there, butyric acid is there. Okay, now when we talk of the different acid, what comes in our mind? Chemistry. Now what is the use of this chemistry over here? See, it has a major use in the field of biology. What is that? Like citric acid, it has been used by the various pharmaceutical industries. Okay, these are been used by the various food industries for processing of the food. It can be used as a food preservative also. Acetic acid, generally we in our houses we are using it in the form of vinegar. Okay, vinegar, it is used as a food preservative as well as it adds the flavor to the food. Then butyric acid. So different types of organic acids are there which have a tremendous use in our daily life. Okay, so these are organic acids. How they are produced? आप citric acid का example ले लीजिए. आप citric acid कहाँ पे होता है? Generally, in case of lemon है या फिर citrus fruit अलग, oranges, okay, मौसम्बी, उसमें क्या होगा? Citric acid मिलेगा आपको सबसे ज़्यादा. बट ऐसा तो नहीं है ना हम लोग पूरे देश भर के जितने भी नींबू है उनको निचोड़ के सिट्रिक एसिड बनाएंगे, नहीं बना सकते। क्यों? क्योंकि जो ह्यूमन डिमांड है या फिर जो पॉपुलेशन की डिमांड है वो पूरी नहीं हो पाएगी, और उसको हमको पूरी करने के लिए क्या करना पड़ेगा, अल्टरनेटिव सोर्स यूज करना पड़ेगा। और ये अल्टरनेटिव सोर्स क्या है कि जैसे हम लोग सिट्रिक एसिड एक माइक्रो ऑर्गेनिज्म से हम प्रोड्यूस कर सकते हैं क्या। अब जैसे कि हम बैक्टीरिया की अगर बात करें, ठीक है, यहाँ पे एक बैक्टीरिया जैसे ज़ैंथोमोनास सिट्री, ओके, या फिर हम फंगस की बात करें एस्परजिलस नाइगर, तो फंगस और बैक्टीरिया क्या कर सकते हैं, दे कैन मल्टीप्लाई विद इन फ्रैक्शन ऑफ मिनट्स। लाइक बैक्टीरिया दे हैव द रेप्लिकेशन टाइम ऑफ 20 मिनट्स, राइट। अब मान लीजिए कोई ऐसा बैक्टीरिया है जो सिट्रिक एसिड प्रोडक्शन कर रहा है और इसको हमने क्या कर लिया, 20 मिनट्स में मल्टीप्लाई कर रहा है ना। तो 20 मिनट्स में भले जो भी हो माइक्रोग्राम या माइक्रो लीटर्स में वो सिट्रिक एसिड प्रोडक्शन कर रहा है। मान लीजिए यहाँ पे आपकी पॉपुलेशन बहुत ज्यादा है, 1000 बैक्टीरिया है, 1000 बैक्टीरिया दे आर मल्टीप्लाइंग विद इन 20 मिनट्स, तो उतने अमाउंट में वो क्या करेंगे, प्रोडक्ट भी बना देंगे, करेक्ट। तो हम लोगों ने क्या किया, साइंटिस्ट ने क्या किया, जैसे उन लोगों ने साइंटिस्ट ने इसको फर्मेंटेशन टेक्नोलॉजी में इंप्लीमेंट कर दिया। और फर्मेंटेशन टेक्नोलॉजी में जैसे इंप्लीमेंट किया तो हुआ क्या, इसका जो मल्टीप्लिकेशन रेट है वो फास्टर हो गया, क्योंकि वहां पे क्या होता है, उसका प्रोडक्ट बनाने के लिए उसे सबस्ट्रेट दिया जाता है, ठीक है। तो जैसे जैसे वहां पे सबस्ट्रेट मिलता है वो प्रोडक्ट में कन्वर्ट करते रहता है उसे, बिकॉज bacteria they have the enzymes, okay, so these enzymes they will convert it that substrate into product। तो यहाँ पे हमको सिट्रिक एसिड जो है वो गैलन्स में, ओके, टन्स में, इतने अमाउंट में एक दिन में मिल सकता है, ओके। सो विद द हेल्प ऑफ फर्मेंटेशन टेक्नोलॉजी, देन एसिटिक एसिड अगेन, ब्यूट्रिक एसिड अगेन, okay, all these acids or organic acids can be produced within a fraction of minutes, okay, so with the help of bacteria, so we have a great use of bacteria in our daily life, okay। So in case of fermented food also we can see pizzas are there, dosas are there, okay, idli is there, all these fermented, even dhokla, okay, all these food products they have been processed with the help of this bacteria, some are processed with the help of some fungi, okay। So microorganisms they are very very useful. Apart from this, many bacteria they are also used, okay, in controlling the pollution. One of the bacteria, okay, is referred as the Pseudomonas putida. Pseudomonas putida. Okay, so this Pseudomonas putida bacteria, it was been discovered, okay, by an Indian scientist called Anand Mohan Chakravarti. And this Anand Mohan Chakravarti, what he have done, he has developed this bacteria into superbug. तो समझ में आ रहा है, तो सुपरबग का मतलब होता है कि जैसे इस बैक्टीरिया में कुछ अलग अलग खूबियां थी, ओके। अब खूबियां क्या होंगी, सो दिस बैक्टीरिया वे यूज्ड टू कंट्रोल ऑयल स्पिल, okay, many years ago, means many not, around 7 to 8 years ago, okay, on the ocean there was a oil spill, and this oil spill, okay, was a major disaster we can say, okay, a kind of disaster or a kind of hazard to the life of the what we say aquatic flora and fauna, okay। So here how to regulate or how to control this oil, as हम लोगों को पता है ऑयल जब मिक्स हो जाता है उसको कैसे हम लोग रिमूव करना डिफिकल्ट हो जाता है, हम क्या कर सकते हैं। So Anand Mohan Chakravarti was working on certain bacteria. These bacteria were capable of surviving in extreme conditions, जैसे कि they were capable of surviving in the condition where the camphor was available. Camphor, it has been isolated from the plant, okay, xanthan, then yes, again on the naphthalene balls, okay, such bacteria he collected, and he collected the plasmid. I have told you about the plasmid, that is extra chromosomal set. Now the plasmid consists of the genes, जैसे कि मान लीजिए some bacteria they are been capable of growing on the camphor. Camphor means what? Kapur, है ना। So this bacteria they have developed a kind of resistance, okay, so they will develop they can consume this oil like this. So what he did, he collected different bacteria of same type and he mixed the DNA of this bacteria into a single bacteria called Pseudomonas putida, and that bacteria was capable of consuming the oil from the water surfaces. So again he was awarded by the a Nobel Prize for such a discovery. Okay, Anand Mohan Chakravarti. And see, it can be acting as a biocontrol agent again. So different bacteria, tremendous bacteria, and their benefits or the beneficial uses are there for the human welfare. Okay, I hope you are getting this. In next part, we will discuss about some few more microorganisms and their uses.